So welcome everyone to this new AgroServ webinar. This is the third in a series of webinars that we will have in the coming months, detailing AgroServ and the research infrastructures that are part of the AgroServ consortium. And it's an opportunity to get to know better the research services that are available in AgroServ, and also to understand a bit more what kind of research you can research services you can access, and in order to advance especially research in agroecology, which is the whole goal surrounding AgroServ and the AgroServ project. So just a few slides to introduce you. We have a new call for projects open with a deadline of January 15th. And we're also very happy to say that next year we will also have at least 2 more calls for projects that are going to be opened. So it's an opportunity for everyone in research, but also everyone that is connected to practitioners and the industry to access the very wide catalog of services that AgroServ offers. And we have a website, agroserv.eu. You can visit and find all the information on how to apply. All the steps are detailed in the website with the guidelines and eligibility criteria that you can find. So I will remind you to the website for all the information that we have. And we also have recordings of past webinars we did with Q&As, which are very specific to the call for projects. Just a practical advice: if you intend to apply, just go to the website and register to the application platform as soon as possible, so that you can get all the information via email, and you can create your own personal platform space to apply and not rush at the last minute to the deadline, which is, as I said, January 15th. If you have any question on the application procedure, I refer you to Dr. Eba Ibrahim. She's our Central Access Manager. You can find her contact also in on our website. And I'm also available, of course, for any kind of question on our project. So let's go to the action, to today's webinar. Today I'm very happy to have representatives from MIRRI and EU-OPENSCREEN, 2 of our research infrastructures, and we have a very big parterre of presenters today, but I'm sure it's going to be a very dynamic and interesting discussion. We start with Robert Harmel, is representing EU-OPENSCREEN, scientific project and industry liaison manager. We have Michel Mistou, is a research director at INRAE, and is also a coordinator in what revolves the research part of the services that are provided from MIRRI in AgroServ. We have Monica Garces Ruiz. She's a postdoctoral researcher at the Laboratory of Mycology at UCLouvain, and she's also an access manager for MIRRI in our project. And then we have in the second section of the webinar, we will have some short keynote presentations from researchers and professionals that are connected to these 2 research infrastructures, and they are Maryline Calonne-Salmon and Anna Martinez, and I will let them be introduced by our representatives from the 2 research infrastructures that can introduce them in a more coherent and complete manner. So without further ado, I will now leave the floor to Robert. He will present EU-OPENSCREEN. So Robert, please go ahead.
Yeah, thank you very much, Daniele, for the introduction. And just share my screen so you can see my slides, right?
Yes.
And good. Yeah, so good afternoon, everyone. Today I want to tell you how EU-OPENSCREEN, as research infrastructure is working together with partner sites and users to discover new bioactive molecules. And so, as you may already know, EU-OPENSCREEN is a leading European research infrastructure for small molecule discovery and also chemical biology. And so our aim is to develop chemical probes for basic and applied research. But many users that come to us, they come usually for developing a drug or to find a small molecule for some agroecological application. We offer technologies for screening. We have also assays that we have at our partner sites. We have a compound collection. We have expertise in medicinal chemistry and chemoproteomics. There is actually bioactivity data available from our database, and we also offer training. And we are open for users from academia and industry, so there's not really a restriction. This is the EU-OPENSCREEN partner site map. So you see here we have now roughly 30 partner sites in 9 countries. The majority of those are screening partner sites. We have 10 medicinal chemistry partner sites and also 3 chemoproteomic sites, which are quite new. So they're not on the map here yet. We're just introducing them. We also have a database host, which is hosting our internal database. So, and for AgroServ specifically, you see here we have the 4 sites within the project, which is the University of Santiago de Compostela. We have Fundación Medina, we have the Polish Academy of Sciences, and all of these are high throughput screening sites. And then also today we have Anna Martinez here from CIB-CSIC, which is a chemistry site within the OpenScreen. So very generally, EU-OPENSCREEN offers 3 types of fields of services. This is, I would say, discovery, optimization, and mechanism of action elucidation. So in discovery, we have a very strong expertise in high throughput screening and also specialized screening. So high throughput screening is usually what you imagine under screening. You have your fluorescent or luminescent readout, and you scan through cell-based or biochemical assays with a lot of compounds. For the specialized screening, it's a bit different. For example, you see displayed here, there's actually a lot of little plants in there. Usually these are more complex models and they give more information, but they also have a little bit lower throughput. So it depends a bit on what you want. And we also have a service for fragment screening. So we have a fragment library actually within the OpenScreen, and fragment screening works a little bit different. And for that, you also need a little bit more follow-up, actually to come from a fragment that you identify in these type of screen to a real small molecule. And we offer all of that. On the optimization side, I think thinking of small molecule optimization, you think about synthesis, probably pops to most of you first in your head, but and we do that a lot. So there's a lot of expertise also in Anna's lab on that, but there's more than just, you know, synthesis and optimization. You also need to optimize molecules against certain applications that you have. This can be, for example, safety and efficacy that I write here, but it can also be, you know, you can also think of bioavailability in plants or into a brain or whatever you are interested in. For the mechanism of action, so when you have your, when you actually find hits, you do not always know how they work. There can be different working principles, especially in cell. If you have a cell-based assay and you don't even know what you're hitting with your small molecule, and so we have actually chemoproteomics analysis available, where you, for example, can develop a probe to pull down the proteins that you're actually hitting on your target, or you can do MS imaging or other type of imaging methods where you can identify, for example, where your compound in a cell is. We also have genetic screening, so you can imagine here that we have that if you have if you basically can basically knock down or knock out certain genes with CRISPR-Cas or small interference RNAs to learn more about the mechanism of action in this way. So zooming in onto AgroServ, we have, as I told you already, 4 partners, and we will hear from Anna more later, so I don't want to spoil that too much. We have these 3 partner sites aside from Anna, which are all high throughput screening partner sites. So they all offer high throughput screening for biochemical and plant cell based assays. And with this usually also comes assay design, adaptation, optimization if needed. With high throughput screening, there's also often mentioned high content screening, which is basically a high throughput screening in an imaging-based type of assay. And so all of these sites come a little bit with a different flavor. So for IBCH Pass, you have there's very strong an image-based analysis and or an image type of assays, and they also offer this AI-assisted data analysis. And they, for example, have also this super resolution microscopy, which is not specifically added into AgroServ. I have to say that's more like a follow-up if you want to find out how your hits work and validate them. And also just for the slide here, so I highlighted a bit in green what is really specifically added into AgroServ, which doesn't mean that you cannot collaborate beyond that with the notes in black here. So for USC, they have a little bit of a different focus. So they're very strong on those high capacity cell culture labs. So they actually have a very capable lab of producing a lot of cells where needed. They also have an HPLC rapid fire system, which can use actually high throughput for MS-based type of assays. And they also have these biophysical assays, like for example, this thermal shift assay, which is not available everywhere. And it's not like a standard screening technique. For Medina, you have a bit of a more really agroecological focus. So they actually have, for example, those biocontrol assays that you can use to screen against phytopathogens, herbicides, or insecticides. And usually, you come with a lot of compounds, and you screen many of those compounds against this different, against one or two assays. They also have assays for biofertilizer assays, and here one of the assets that they also have internally is they have a huge library of microbial strains and natural product extracts, which can be utilized for all kinds of applications. And they also have hit profiling. For example, they do a lot of metabolomics at Medina. So one key asset within the OpenScreen is actually the OpenScreen libraries. We have 3 different types of libraries: that the European Chemical Biology Library called ECBL, we have the Academic Compound Library, and the Fragment Library. The European Chemical Biology Library is a library that was designed from commercial compounds, but this unique setting is only available within the OpenScreen, and it's a diversity library, right? So this is really the ones that we use for high throughput screening. And we believe it's a very good library, especially for screening purposes. There's a small subtype called the bioactive. So here's actually, we know what targets are hit by these compounds. A second library that we have is the Academic Compound Library. Here, chemists can actually submit compounds to the OpenScreen, and they are made available to the screening community. So these can be really novel compounds or compounds that you cannot get anywhere, and you can screen them actually, for example, against your cell assay. And then you have the Fragment Library, which will be used for fragment screening, which just works a little bit different than classical screening. So I'm almost at the end. So this is the European Chemical Biology database. So all the data that we produce within the OpenScreen is actually supposed to end up within this library, at least for the screening data. And here we disclose really the compounds, the targets, and also the assays after a certain embargo period. So we're really trying to make the data from the screening available. And I'm talking about positive and negative data. So also if your compound doesn't hit in a certain assay, will be documented here. And that helps the whole scientific community and also all the people that want to actually work with our compounds in the future. Of course, there's a little bit of a time that you get to publish or patent your results, before we publish that. So actually, we're at the end of the presentation from my side. I just want to highlight one more thing, because I said in the beginning, we also do training, and there's actually next week, there's a large training school from the OpenScreen, and it's for free. And if you really want to work with us, it's really helpful to understand how you can design your assay, how you can provide, and learn what is important to have your high throughput screening assay, maybe also medicinal chemistry aspects and so on. So there's a very good opportunity. You can just sign up, and there's no other strings attached. So that's it. Very much. Thank you for listening. I'm happy to take any questions.
Thank you, Robert. Thanks a lot for the very quick and comprehensive presentation of your OpenScreen. Yeah, of course, if there are questions, we will have a short Q&A at the end of the webinar. So I invite everyone to take notes, and if you have questions, to bring them at the end. So let's move on. I will leave the floor to Michel-Yves, who is going to present to you MIRRI. So Michel-Yves, you can start your presentation, and I leave you the floor.
Is that okay for the sharing?
Yes,
No.
if you want to enter the presentation mode, yeah, that's great.
Okay, thank you, Daniele.
Thank you.
So I'm going to present you as fast as possible the MIRRI-ERIC infrastructure, and maybe I will focus on the of its involvement in AgroServ project, and so how MIRRI-ERIC can serve the agroecological transition. So the outline of the presentation would be, so it's quite a challenge, so a rapid overview of MIRRI-ERIC, and then of some of the services we are proposing in the AgroServ framework. So some key figures about MIRRI-ERIC. So it is MIRRI-ERIC. MIRRI obtained the ERIC status in 2 years ago. And we are actually 6 countries are full member of MIRRI. The figure on the right is not fully updated. I'm sorry for Greece, but Greece has joined MIRRI a few months ago, and we expect to have 2 additional members in 2025. And there is Romania, which is one observer. MIRRI-ERIC gather about around 50 microbial resources centers, and so can propose on its catalog around 400,000 microbial resources. So it is viruses, bacteria, fungi, plasmids. Its activity on average it's around 6,000 users per year, and 20,000 biological samples are provided every year to the research or industrial partners. The activity, there are 7 strategic research areas which have been defined for MIRRI-ERIC activities. So research in the mechanism of infectious diseases. Of course, also the research on therapeutic intervention on these infectious diseases, especially, for example, research in the field of antimicrobial and antibiotic resistance. The food and feed domain, with, for example, the research on food fermentation, soil and crops, because there is, as you probably know, a colossal biodiversity, microbial biodiversity in soil and associated to plants. And these microbial communities, they are very important for soil and plant health. The 5th domain is environment. And we have, for example, many partners in MIRRI which are involved in the use of microbial resources for bioremediation. Then biotechnology, with, of course, some activities, many activities in the biofuel, new materials, bioconversion, etc. And in more fundamental research on microbial biodiversity, which is expected to be huge and highly underestimated at the time. So in the frame of the AgroServ project, MIRRI, which is a small partners of AgroServ, propose 11 type of services to support so the research in that domain. So they can be roughly divided in 3 main categories. First, the supply of microbiological resource, and of course, the associated expertise for handling, conservation, propagation, and implementation of screening assays, and formulation of microorganisms. The second category is characterization assays, test assays, MIRRI or users samples. So we have been contacted for example, testing for plant growth promoting activities, anti-infectious activities against plant pathogens, fermentative bacteria and yeast for the agro-food sectors, or characterization of microbiota. And the third type of services provided by MIRRI is training activity, and I think that this will be developed by Monica after me. So category 1, so the provision of microbiological resource. So when it comes to biological resource, if we have an oversimplified vision of it, we can say that it can be divided on on 2 categories: the good guys and the bad guys. So the good side of microbes is, of course, that they are, as I told, associated to plant and soil health, for example, and so they can be used as biofertilizers, biocontrol agents, bioprotection, biostimulation, bioremediation, and in the food domains for the fermentation activity. And we have, for example, here the examples of mycorrhizal fungi, which can be provided by the University of the Louvain, which are very important for providing important nutrient to plant, which are essential for plant growth. And on the other side, we have the bad guys, the pathogens, which are of course yes, problematic for agriculture in general, and agroecology also must be protected against this. And MIRRI is providing the pathogen also for research on this activity, and also some services to fight against its pathogens. And concerning this, the second category of services of MIRRI, so the providing more integrated services. Yeah, I give one example for the characterization of microbial resources. It's some kind of screening test for antimicrobial activity. So the TNA users, potential users of the service, can provide molecules or plant extracts or any kind of extract he thinks, or he put the hypothesis they can have some interesting biological activities, and we can propose some selection of biological target on which we can test the biological activities of this extract through different kind of methods that can be implemented in our laboratories. A second type of example is characterization of plant-associated microbiota. So we can propose services to characterize both on the metabolic activities and the genetic composition of microbiota, which can be extracted from different plant organs like leaf, root, or seed. We have some protocols for performing the extraction of the microbial communities, and then to characterize them, this community metabolically and genetically. And so it becomes possible to when you associate, the taxonomic composition of the sample can be determined, and of course we have also this conservation options that is proposed by the Microbial Resource Center. And you can then have some integrated this type of analysis can be used to perform some comparative analysis of microbiota isolated from different sources or different plants which have been grown in different conditions. And for example, this services is implemented in one AgroServ project, which is dealing with the consequences of soil amendment on microbial community composition. So I will stop now. Just one concluding remark. Most of the MIRRI, and I will take the opportunity of the presence of EU-OPENSCREEN to make this this conclusive remark that most of the MIRRI assays which are proposed actually are not carried out at very high throughput, and so it might be interesting to discuss with EU-OPENSCREEN the opportunity of developing in that direction, given all the resources and expertise they have in this high throughput assays. Okay, thank you for your attention.
Thank you, Michel-Yves. It's very interesting. You started to make this connection, and I think hopefully we can have a bit of a discussion maybe at the end of this session. I think it might be very interesting for anyone who is attending. Thank you. So Monica, maybe I leave you the floor to kind of introduce our first. Before introducing our first, let's say, keynote speaker, to go a little bit more in depth into the research. I know you also wanted to add something regarding the TNA offer, so please go ahead, Monica, and the floor is yours. You're muted at the moment.
Can you hear me now?
Yes.
Yes.
Perfect.
Thank you, Daniele, for this organization of this webinar. Yes, I wanted just to inform the participants that we have 4 services in the laboratory that they can be used. And so the first one is the identification, preservation of fungi, focusing on agroecology. And there are some fungi that can be used as biocontrollers and biostimulants, as Michel said. And here we have the opportunity to identify this kind of microorganism, and the people can be learn how to preserve these microorganisms. The second service that we have is to have access to different fungal strains that we have in our collection in the laboratory. The third service is a 5-day in vitro culture, a training on in vitro culture of arbuscular mycorrhizal fungi. In this training, the participants will learn about the techniques that we use in our laboratory. And the fourth service that we are offering is the formulation of biocontrol of fungal agents. And here the idea is that you learn how to entrap these fungi in microbeads that can be used in agroecology. And for this, my colleague Maryline, she will present you a case of study about this subject, microbiomes. And Maryline, she is a senior researcher at the Laboratory of Mycology for 10 years, and with a PhD in mycology and environment. And she is involved in various projects, mainly focused on the biocontrol of cross disease and pests, phyto remediation, polluted soils, and microbial interactions. So I give the floor to Maryline.
Thank you, Monica. We just have to exchange the field, but we are in the same room. So just 1 second. I'm gonna share my screen. Okay.
Yeah, we can see you. Perfect.
Okay, you see my screen?
Yeah, perfect.
Yes. Okay, great. Well, thank you, Monica. I'm very pleased to present to you today how entrapped beneficial microorganisms improve yield and plant resistance against pathogen infection, with a case study on the potato yield in field and on the control of potato lead blight in greenhouses conditions. As you probably know, potato is one of the most important food crop in the world, and unfortunately, it is a highly susceptible crop to pest attack and disease infection. One of the most devastating diseases is the lead blight caused by Phytophthora infestans, a nomicide present in every country where potato is cultivated, as visible in orange on the map, so quite everywhere. In field, the lead blight is identified by brown lesions on leaves and stem, and tubers could also develop black spot with a higher risk of second contamination by bacterial rot, making them thus unmarketable. This pathogen could destroy a potato field within a few days, and global loss represents each year 6 billion sterling. The control of the lead blight requires each year between 8 to, most of the time, 20 fungicidal treatments according to the weather conditions, with chemical pesticide or copper, which is authorized in organic agriculture. Currently in Belgium, to control lead blight, 97 chemical molecules are authorized versus only 5 biological products, mainly plant extract and kosoga, and none are composed of microorganisms. Despite their effectiveness, the application of chemicals and copper on soil can have adverse effects on human health and cause pollution issues in the soil, thus affecting the microflora, inducing shift in the microbial communities, in addition to the development of resistance by the target pathogens. To solve these issues, we need to develop alternatives. Among them, microorganisms as fungi are, however, an inexhaustible supply of solutions to control crop disease. So far, many antagonistic fungi, as well as endophytic, symbiotic fungi, have been investigated. They can indeed produce antibiotics and antifungal molecules. They can stimulate the plant defenses. They can compete with undesirable pathogens and pests, and for some, they can parasite fungi. This includes generally fungi as Trichoderma, Fusarium, Penicillium, Aspergillus, etc. Among symbiotic fungi, arbuscular mycorrhizal fungi, or AMF, are known to improve host plant nutrition and increase plant growth. For people who don't know them, after germination of a spore, the AMF develop in the root cortex of the majority of crop plants, where they form arbuscules, the structure involved in mineral element and signal exchange between the fungi and the plant. They also form an extensive mycelial network in the soil where they uptake mineral elements and water to transfer to the host. And among other benefits, they also stimulate the defense of the host plant against the pathogen infection. So based on these benefits provided by fungi, in the laboratory of mycology, we research new fungal-based solutions for a more sustainable agri-food system and their formulation for an easy application in field. So I'm going to show you some results we have obtained, starting with a first experiment conducted in greenhouse, in which we investigated the potential of fungal extract combined with AMF to control lead blight on potato. So for this, we first selected 150 fungal candidates from the scientific literature, which were provided by the Mycotech of UCLouvain. You can see here the website. For your information, the collection holds over 30,000 strains of filamentous fungi and yeast-like fungi, representing more than 5,000 species. So if you are interested, please visit our website. So the fungal molecules were extracted from the fungi, selected fungi, and were tested against Phytophthora infestant sporangia in microplate wells, and our results showed the total inhibition of the sporangial germination with 17 of these fungal extracts. In the second experiment, the 17 extracts were applied on a growing mycelium of Phytophthora infestans in a Petri plate. So from this screening and other pre-tests we conducted, Trichoderma virens and UCL18139 was the best candidate. It was thus tested in planta with the AMF Rhizophagus irregularis and UCL41833. And they were both entrapped in alginate microbeads as visible on the picture. You have to know that the formulation of microorganisms in microbead, in alginate microbead, is a biotechnology which allows the preservation of fungi, of AMF, of bacteria in good conditions, and increases the persistence of molecules over time. It thus offers an excellent protection of the microorganisms and molecules against biotic and abiotic stress in field, in addition to be biodegradable, non-toxic, non-pollutant, and very importantly, microbeads are compatible with the agricultural machinery. For our experiment in greenhouse, AMF spores produced in vitro were collected as well as the extracts of Trichoderma virens, and they were both entrapped into alginate microbeads using a peristaltic pump as visible on the picture for an easy and uniform application in the substrate of the pots which served for the potato cultivation in greenhouse. We can see on the picture here the germination of the spores of Rhizophagus irregularis and the subsequent growth of ivy outside of the alginate microbead. You can see them with the arrows. And this demonstrates, one, the capacity of the fungus to survive and germinate in microbeads, and two, to grow outside to further colonize the host plant, without here any impact of the molecules produced by Trichoderma virens. At planting, alginate microbeads entrapping both Rhizophagus irregularis and extracts of Trichoderma virens were distributed around the tubers. One treatment also consisted of one spray of extract of Trichoderma virens on the leaves before or after infection with the pathogen. Other tubers were planted without any treatment. After 8 weeks of growth, sporangia of the pathogen were inoculated on the plants, but the negative control, of course. Another positive control and a fungicidal control, which consisted of a foliar application of mancozeb, were included to this assay. And 2 weeks later, the leaf infection by the pathogen was recorded. Our results showed a significant reduction of the foliar symptoms of leaf blight on plants inoculated with the AMF and the extracts of Trichoderma virens. As visible on the graph, the level of infection was as low as the fungicidal control, which is a really good result. This also demonstrates the systemic protection conferred in the aerial part of the plant by the fungi and molecules which were inoculated at the root level. Curiously, we can see that all plants sprayed with the extract of Trichoderma virens developed foliar symptoms equivalent to those of the positive control treatment. To explain these results, we cannot exclude that the extracts containing enzymes, acyclic enzymes, and for instance, affecting the potato leaves and increasing their susceptibility to infection by the pathogen. In another experiment led in field, this time, we investigated the potential of combination of fungi to improve potato yield. We indeed evaluated the capacity of Rhizophagus irregularis and UCL41833 combined with Trichoderma arzanium and UCL29707, both key soils microorganisms and known to increase plant growth. So we evaluated their capacity here for this assay to improve the potato yield in field conditions. For this, we applied the microorganisms entrapped in microbeads to a cover crop, Medicago sativa, preceding the potato crop. Or we directly inoculated the potato at planting. Two controls were included and consisted in the absence of any treatment preceding potato crop or only the cover crop alone with Medicago sativa before the potato planting. For this, after production of the AM fungal inoculum in vivo, this time, using pod culture of maize as host, and collection of Trichoderma spores from the in vitro culture, we entrapped the microorganisms together, as explained earlier, using a peristaltic pump to obtain regular and uniform beads. And these beads, as I said, are compatible with the machinery, and they were applied in field using the agricultural machinery, for instance, here, a grain seeder, in which we can see the seeds in yellow and the microbeads in white, which are quite the same size, so they can be applied at the same time. And the size of the beads can indeed be adjusted according to your needs. For this experiment, we first tilled the soil and sown the cover crop with the alginate microbeads, containing both Rhizophagus singularis and Trichoderma arzenium in August to see the first stage of developmental stage around October. Then, in spring, the cover crop was cut and the soil tilled before the plantation of the potato crop. Our treatments also consisted of the direct inoculation of potato with the microorganisms at planting. We can see some beads around the tuber. At harvest, we measured that the best agricultural practice to increase potato yield in this assay was the inoculation with both microorganisms of a cover crop preceding the potato crop, as visible on this table, with a significant increase of weight of tuber, as well as a higher production of tubers per plant for this treatment in comparison with the other ones. The inoculation via cover crops also seems to be a more efficient strategy as compared to the direct inoculation at potato planting. Probably the cover crop increased the natural and or the inoculated population of beneficial microorganisms in the soil during the winter before the potato planting. In conclusion, we noticed that fungi can be a source of solution to improve crop yield and control plant diseases, thus allowing a possible reduction of the chemical inputs and pesticides for more sustainable agriculture. In addition, their formulation in alginate microbeads is an efficient tool to preserve fungi and molecules they produce and for a facilitated application in field. As we all know, further researches still remain to be conducted to improve the fungal efficacy in various conditions in field, various soil conditions, various climatic conditions, etc. So a lot of work remains to be conducted. Interested in our research on fungi and alginate microbeads, so please have a look, please read these papers and numerous others we have published in the Laboratory of Mycology the last years. And finally, as Monica mentioned, if you are interested in learning how to make the microbeads, how to produce the culture of AMF in vitro and in vivo, we organize each year the international training on in vitro culture of AMF. The next one is planned the first week of June and is given each year, so don't hesitate to apply. And if you are interested, please visit our website, mycorrhiza.be. Thank you for your attention and see you later for possible questions.
Thank you, Maryline. Thanks a lot for the presentation. I just want to, Robert is here. I leave you the floor right away.
Yeah, great. So I just want to briefly introduce Ana Martinez. So Ana Martinez is, together with Carmen Gil, leading the group of translational medicinal and biological chemistry at the CIB-CSIC in Madrid. And she's a very active and a very active member of EU-OPENSCREEN network and also a certified chemistry partner site. And she focuses on innovative small molecules with novel mechanisms, for example, for infectious diseases and neurodegenerative diseases, but also, of course, have other interests. And she provides, for example, chemistry, but also classical chemistry, but also computational chemistry. Yeah, let's go, Anna.
Okay, so thank you so much, Robert. And so, as he mentioned, we are the Translational Medicinal and Biological Chemistry Laboratory. We are based in the Center for Biological Research that belong to the High Research Superior Council of Spain, and we are based in Madrid. So this is the presentation of our team. This is our center, some of our laboratories, and as
Anna, we can only see your first slide, I think. So.
Wow. Wait a minute. So you cannot see the next slide?
No, we just saw your first slide in non-presentation mode.
Now I have nothing sharing, so.
Okay, do you want me to share? I can share.
Share you, and
Okay.
I said.
So now you
So
should
this is the first one. We are in the second.
Yes. So, and I will. Okay, now.
Okay, so this is our center. Our main laboratories are synthetic ones, but also we have cheminformatics hardware and software to perform both in design and heat-to-lead optimization of our molecules. And also we have a small chemical library in-house, which is known as the MBC library, with the compounds that we have synthesized in all of our experience. Here you can find the photos of my colleague Carmen Gil and myself, who works together in these chemistry sites of European OpenScreen. So, although our main experience is based in drug discovery, obviously we have the same methodology we can apply to any molecule that have a biological activity. And so in the next one, you have the methodology that we use, which is called chemical genetics, and we use a direct forward chemical genetics and also the reverse chemical genetics. The main difference is the starting point. In chemical genetics, we start testing molecules in a phenotype in order to select a phenotype of interest. And afterward, this molecule that have produced this phenotype is investigated in order to identify some proteins that are able to bind to this molecule and responsible for this phenotype. In the reverse chemical genetics, is just in the other way. So the first point is to select the protein of interest, screen different chemical libraries, and so when we have one molecule with good binding, observe the phenotype that we would like to produce. So in the next one, we will see how this chemical genetic approach has been used to identify some molecules that are able to increase the embryogenesis induction in different in vitro cultures of different species of plants. So initially we have selected very different chemical compounds that the only point of common is that they are JSK3 inhibitors of mammal JSK3, but we tested in 4 different type of in vitro cultures and different type of plants, including some trees as quercus and pine. and we increase the regeneration or enhance the number of embryos. So that may be applied to recalcitrant species. In the next one, we can show another kind of inhibitors that we have used, which is LRRK2 inhibitors. Typically, this kind of compounds are being developed in the medical science for treating Parkinson, but we know that they have neurogenesis properties in human cells, and we apply also to different in vitro cultures of embryos in different conditions, and again we obtain very good results. So as a recapitulation of this couple of slides, we can say that using small cell permeable synthetic molecules, we have the opportunity to enhance in vitro plant regeneration yield, and so chemical modulation of different kinase activity by small molecules can be used to overcome pure embryogenesis induction and cell proliferation in different plant tissue culture. So we have filed a patent, and now this technology is worldwide use, and you have different companies that now are using these compounds to increase the yield of different kind of culture. So in the next one, we are going to show you some results using the reverse chemical genetic. As I mentioned before, we can use any protein of interest that you can have from your plants or your animals or microorganisms that infect your plants or your animals, and we don't know, it is not important in this step if we know or not the three-dimensional structure. So we need to use chemical libraries, and as being European OpenScreen member of AgroServ, we can use the European Chemical Biology Library that have more than 100,000 molecules, as Robert mentioned initially. Also, we can test different libraries of different approval compounds, such as FDA or our own library. And so we perform virtual screening of this chemical library to prioritize the compounds in order to observe a special phenotype. So in the next slide, we will present you a working case that we are working now. And in that case, the researchers came to us asking for inhibitors of an arginine kinase from mites as pan-allergen target. So firstly, we built the three-dimensional structure of this arginine kinase using different software. And after we performed virtual screening using our MBC library, which is our in-house library, we select less than 20 compounds, and we perform phenotypic screening by adding these compounds in the mite diet. And so now we have new acaricide compounds, and the project is ongoing. In the next one, we can show another method, because when we perform rational molecular or drug design, we consider the three-dimensional structure of the protein or the macromolecule where our small molecule interacts, and also the chemical structure, three-dimensional structure of our molecules. But we can present different situations that neither the compound nor the protein is known, or only the compounds, only the protein, of both cases. In the next slide, we have shown just now different examples, starting by phenotypic screening, in where it is not important know anything about previous compounds of previous proteins. And so the second approach in where we have starting by building the three-dimensional structure of a required protein. But now we have, well, if we know both structures, what we can do in our laboratories, for example, if some in our laboratories you have identified a bioenergetic compound, a fertilizer or insecticide compound, and you need to improve some physicochemical characteristic as increased solubility or permeability or something like that, we can start using these techniques. And so in the next slide, I'm going to show you another approach that we can use also. That is when only what we know is the structure of different compounds that are effective but has several side effects or several problems, like for example, fenbendazole, which is an antihelmintic used in different fish culture. And we need better antihelmintic agents in order to preserve environment and human health. And in that case, what we have done is starting from the chemical structure of fenbendazole and using different topological indices, such as Tanimoto, Buser, or Cousin, define the fingerprint of this compound, and with the fingerprint search in a chemical library as it is MBC1. And so in that case, we identify compounds and we testing in vitro cultures of these specific nematodes. And now we have new antihelmintic agents with better properties to be optimized. The project is ongoing. As you can see, the different chemical structures are different to the first one, and we need to explore further characteristics. So in the next one and the last one, the take message home is that in fact the life started in small molecules. From then we have all the development for the tree of life, where microorganisms, plants, and animals, inferior and superior, are developed. And after that, small molecules can be actioned from all the life, animal and vegetal, and we have many common cellular signal in common node. So our team is ready to offer to you help in finding molecules that makes more effective all the biological process you are involved in. And that's all. The last one is only to thank you and for your attention and to give our mails. That's all. Thank you. Open to the questions.
Okay, thank you, Anna, and also thank you. I would like to also thank Maryline for her presentation again. I think these two short presentations highlighted very well the kind of opportunities that you can have accessing the services provided by AgroServ. I can now open the floor for any question from the audience if we have some. But maybe I can start with maybe a suggestion to Robert, to Monica, to Michel-Yves, taking up maybe the suggestion that Michel-Yves made at the end of his short presentation of MIRRI, like how can we enhance a little bit the interaction between research infrastructures? Because I think these two presentations from Anna and from Maryline highlighted a little bit the fact that at the end of the day, many topics are very interconnected. And so the idea of having EU-OPENSCREEN and MIRRI together, it was a bit kind of a way to suggest this interaction to potential users that might want to carry on their research. So maybe, I don't know if Robert, you wanna take on the suggestion from Michel-Yves, and if you want to add something, and then we can have a round table. I don't know.
I think that suggestion was very interesting. I think we are trying to always look left and right of what we are usually doing, and we never get to a spectrum where we actually, I mean, sometimes we identify things, but it's good to hear that kinds from Yves to that this opportunity is there and that this is actually highlighted to us. And I think it totally makes sense, right? So there's a lot of essays out there that we have the expertise to kind of automate them to have them for higher throughput. Will not all be high throughput screening. So I think it makes total sense to like discuss this and work on this. How do we enhance this in the future? I think Eve and I need to sit down, talk about it, and maybe we can even do this within an AgroServ during a workshop that we're organizing in parallel or something. But I think it's something that also will take a bit of time to align here. But it sounds like a great opportunity. I really like that idea. Yeah.
Okay, and if anyone wants to add something, I think we can reach the conclusion of this intense hour. But anyway, yes, to add to Robert, maybe I can also say that AgroServ can serve a little bit as a starting point, since the project will come to an end in 3 years, 2-3 years. So maybe it's also the opportunity to start to draft up a little bit a legacy of what's going to happen afterwards. Michel, go ahead.
No, yes, I think it was a lot of information to process today with the presentation of Robert and the last one of Anna. So I think it could be worth having kind of EU OpenScreen, MIRRI, little workshop or meeting together, so that we can maybe present in a little bit more details what we are doing. And I have a question for Robert, who mentioned as one of the EU OpenScreen partner, Medina, who seem to have quite a large collection of microorganisms. And maybe can you tell us a little bit more about that? Is this a catalog which is open, or maybe they already have some good expertise in the screening of microorganisms?
I see Anna nodding, and I think she even knows more than I do. But I think overall they are open to collaborate on those. So I think this is a resource that they are using to collaborate with people. So it's not just an internal asset. And they're very experienced in like screening and these type of. But I really would suggest we need to talk to Olga Genuit directly, because I think there's a lot more behind this than what I showed today. And I think it's always best to directly talk with a person responsible, because there's so much detail that I might not interpret in the right way, let's say. Yeah. I don't know, Anna, do you maybe want to add something on that note?
No, yes, in fact, in Medina, they are a team with a high level of expertise in isolate different compounds from different microorganisms. And so they have a vast expertise in move to a high throughput screening, the evaluation of these microorganisms. And so without any doubt, they can help you in this task. And so, yeah, I am also a believer that our molecules and the molecules that are in the ECVL library will have value for the microorganism, but also our idea today is that all AgroServ partners try to find in the technologies that we have available a good instrument for collaboration and mainly for increase the results of the research, no?
Yes, definitely. I think it's very appealing for us. And what is the name you mentioned of the people in charge of Medina?
There's several people, but it's Olga Genuit is the director there, and I can just connect you to her. That's we can.
Yes.
Let's
Would be
follow up
nice.
after then. Yeah.
Okay.
Okay, well, I'm very happy that connections start to be made, and also something that maybe people outside can see happening, and they can maybe future users can be the ones that push forward this kind of connections, hopefully, and since we require transdisciplinarity and we require to actually connect different research infrastructures. So I would like to thank you all for participating to this webinar. I just remind everyone that the recording of the webinar will be published, and it will be available on our website. So thank you, everyone. Have a good day and see you soon.
Thank you, Daniele, for the organization.
Thank you very much.
Bye, everyone.
Bye.
Bye. Bye.
Bye.